Assalamualaikum and good day everyone. Today we will discuss on transport across membrane, specifically passive transports. But before we proceed, I would like to give credits to Madam Nur Fazlini for her expertise, simplified and prepared the notes which were taken from our main reference, Campbell and Rees, Biology, A Global Approach, 11th edition. So this is the last subtopic of chapter 2, cell transport, which actually we are referring on the transports across the plasma membrane. As all of you aware, the plasma membrane is very selective. So at the end of this lesson, you should be able to explain the various transport mechanisms across the membrane, which include passive and active transport. Passive transport could be divided into three, which are simple diffusion, facilitated diffusion, and osmosis. Whereas active transport will include sodium-potassium pump and bulk transport. Bulk transport is actually can later be divided into endocytosis and exocytosis. So let's have a look at the passive transport first. Passive means this kind of mechanisms, these kinds of transport does not require ATP. Okay, does not require ATP. ATP should be written capital letters. Okay, ATP. Right, that is the first characteristic of passive transport. And it is down concentration gradient, meaning that the substance moves from the higher concentration region towards the lower concentration region. And this passive transport can be divided into three types, which are simple diffusion, facilitated diffusion, and osmosis. Let's have a look at the simple diffusion first. This is the movement of substance across the plasma membrane from a region of higher concentration to a lower concentration. So meaning that the substance, for example, the molecules moving down concentration gradient, region. Okay, the keyword there is region. So what are the substance? The substance could be molecule, ion, or atoms. The very easy example is the gases, and then the lipid-soluble molecules. Second is facilitated diffusion. Okay, but this is when we mention facilitated, meaning that this kind of transport mechanism requires something to facilitate the diffusion, because it is not simple diffusion. So what kind of things or structure that facilitate or assist the diffusion? Okay, so this facilitated diffusion is helped or assisted by transport protein. You must remember, okay, when we mention transport protein, it might be carrier protein or channel protein. They play a different role. Okay, so molecules that can pass through this kind of facilitated diffusion are ions, polar molecule, large molecule, and hydrophilic molecule. So as I mentioned earlier, so the transport proteins could be channel protein or carrier protein. So let's have a look at the channel protein first. Channel protein, okay, where the protein provide a passage which allow specific molecule or ion to cross the membrane. So what are the substance that can pass through this channel protein? Example is ions. Sodium ions Na+, or potassium ion K+, right? Another type of transport proteins is carrier protein. This kind of protein change its shape and help to translocate the solute binding sites across the membrane, change shape triggered by binding and releasing of transport molecule. Alright, so a very good example for carrier protein is the protein that help to transport glucose, because a glucose is a polar large molecule. As teacher mentioned earlier, there are two types of proteins. There are two types of transport proteins that assist the facilitated diffusion. Let's have a look at the channel protein. Channel protein is quite simple compared to the carrier protein. We take sodium ions as example. So let's have a look at the diagram on the right. Okay, sodium ions are at higher concentration at the extracellular fluid compared to the sodium ion concentration in the cytoplasm. Therefore, the sodium ions will move along the channel protein from the extracellular fluid into the cytoplasm. Whereas for the carrier protein, it involves the changing of the shape of the protein. Okay, let's take glucose as a good example there. The glucose concentration is higher at the extracellular fluid compared to the glucose concentration in the cytoplasm. So, once the glucose bind to the binding site of the carrier protein, it will trigger the changing of the protein. Okay, therefore it will lead to the release of the glucose to the cytoplasm. This is the third type of passive transport, which is osmosis. Osmosis is the net movement of water molecules from a region of higher water potential to a region of lower water potential through a partially or semi-permeable membrane. So in this osmosis, we have to understand two concepts, which are to determine the water potential in a cell, whether it is in animal cell or in plant cell. So what is water potential? Water potential is the term given to the tendency for water molecules to enter or leave the solution by osmosis, meaning that the keyword there is the tendency of movement of water molecules. Pure water has the highest water potential, which is 0. So if we dissolve solute molecules into pure water, it will reduce the concentration of water molecules. So pure water molecules plus solute will give negative water potential. Therefore, water diffuses from region of higher water potential, which is less negative or 0 volume, to a region of lower water potential, which is more negative value. So bear in mind that pure water is 0 megapascal. Okay, so if we put solute, for example, if we are preparing a glucose solution, so when we add glucose in the pure water, so the water potential will reduce, so it will give a negative value for the water potential when we add glucose in the pure water. So what is solid potential? It is a force of attraction towards water molecule caused by dissolved substances in the solution. Solid potential is negative component of water potential due to the presence of solid molecule. The more negative the solid potential, the higher number of solute in the solution. Solid potential is inversely proportional to water potential ataupun berkadar songsang dengan water potential. Let's have a look at this diagram. Okay, so the solid potential in the cell, okay, is lower than the solid potential outside of the cell, because in the cell, okay, there are only about 6 sucrose molecules. Therefore, since the concentration or the amount of sucrose in the cell is less compared to the amount of sucrose molecules outside of the cell, therefore, the solid potential in the cell is less compared to the outside or in the solution outside of the cell. Meaning that the cell there, the water potential is higher compared to the water potential outside of the cell. So what is pressure potential? Pressure potential is the physical forces that act on a cell. Pressure potential is generally positive and it is only applicable to plant cell due to presence of cellulose cell wall. Animal cell has no pressure potential. You have to remember that because animal cell does not have cell wall. So this is the equation that you have to understand to determine the water potential. Psi, okay, the water potential or the water potential of the cell in the cell, okay, represented by symbol psi. Okay, psi equal to solid potential plus pressure potential. So if I given with, okay, here the core pressure here is the pressure potential is 0.5 megapascal. So if there is pressure potential, meaning that we are referring to plant cell. Okay. So let's have a look at this diagram. Right? Given here, the pressure potential is 0.5 megapascal and solid potential is negative 0.2 megapascal. So using the given formula, okay, which is psi equal to solid potential plus pressure potential, therefore the water potential in the plant cell is negative 0.2 plus 0.5 megapascal, making it 0.3 megapascal. So why is the water potential in the solution is negative 0.2 megapascal? Because the solution outside of the plant cell does not have pressure potential. So we ignore the pressure potential, which is 0.5 megapascal in the plant cell. Therefore, the water potential in the solution is negative 0.2 megapascal. So this is the second concept which I mentioned earlier. Okay, so the three types of the solution are hypertonic, isotonic, and hypotonic. So what is hypertonic solution? Hypertonic solution is low water potential relative to another solution. Example, the cell's cytoplasm. So if a cell placed or put in a hypertonic solution, remember hypertonic solution is a solution that has less water molecules. Therefore, water will diffuse or move out of the cell, causing the cell to shrink and shrivel. Water diffuse or move out of the cell via osmosis. You have to mention that. Whenever we mention the movement of water, it is in a package, meaning that you have to mention the water move or diffuse via osmosis. So when the water diffuse or move out of the cell, it will cause the cell to shrink and shrivel. So in plant cell, the process is called plasmolysis. Process name is plasmolysis, but the plant cell, okay, the behavior or the verb for the plant cell is plasmolyzed. Understand, this is quite a different term. One is plasmolysis refer to the process name. The condition of the plant cell is plasmolyzed. However, for the animal cell, the process of water diffuse or move out of the animal cell is crenation process. Okay, the name of the process is crenation. So what happened to the animal cell? What is the condition of the animal cell after the water diffuse or move out from the animal cell? So the animal cell will become crenated, shrink and shrivel. So you have to to bear in mind the different process names for plant cell and animal cell. So the second type of solution is hypotonic. Hypotonic is a condition where high water potential in the solution compared to the other solution. For example, when a cell is placed in a hypotonic solution, meaning that the cell has less water molecule, but the solution has more water molecule. So water will diffuse or move into the cell via osmosis. Remember, you have to mention the name of the osmosis, causing the cell to swell and possibly burst. The plant cell becomes turgid. Turgid is the condition when the plant cell receive water from the hypotonic solution. Okay, the condition of the plant cell is turgid. But for the animal cell, okay, the process is called hemolysis or lysis. But animal cell will swell and hemolyzed or burst because animal cell does not have cell wall. The third type of solution is isotonic, where the condition is water potential of one solution is the same as another solution. The cell place, for example, when the cell placed in isotonic solution, water still diffuse, move into and out of the cell at the same rate, and the cell does not change in shape. We cannot say that there is no movement of water molecule at all. No, the movement of water molecule still occur, but at the same rate. So what happened? Okay, plant cell will become flaccid, but animal cell will experience no changes. Now let's have a look at this beautiful diagram. These are two different conditions when animal cell is put. One is in hypertonic solution and another one in hypotonic solution. Let's have a look at the animal cell when it is placed in distilled water. Remember, distilled water has no solute, therefore the water potential is zero, which is equivalent to pure water. So what happened is the water molecules from the distilled water will diffuse into the animal cell and make or resulting in the animal cells become swell and burst because animal cell does not have cell wall. However, when the animal cell is placed in a concentrated salt solution, okay, so meaning that in the salt solution, the water molecule is less compared to the water molecule in the animal cell. It makes the animal cell, the water molecule in the animal cells will diffuse or move out from the animal cell to the salt, the concentrated salt solution, making the animal cells shrink and shrivel. Now let's have a look at this beautiful diagram where the plant cell is placed in two different solutions. Okay, so have a look at the plant cell when it is placed in distilled water. Distilled water is a pure water where the water molecules is very high compared to the water molecules in the cell. Therefore, the water molecules from the distilled water will move into the cell or the plant cell via osmosis. It will make the cell or the plant cell stiffens but generally retains its shape. Whereas, if a plant cell is placed in a hypertonic solution, where this solution is, for example, a concentrated salt solution, this hypertonic solution has less water molecule compared to the water molecules in the plant cell. Therefore, the water molecule in the plant cell will move out or diffuse out to the concentrated salt solution or the hypertonic solution via osmosis. So this will make the cell body shrinks and pulls away from the cell wall.